Okay, so let's now come to something called brown and white dwarfs. Now, there are several types of dwarf stars. There are brown dwarfs, there are white dwarfs, there are red dwarfs, and there is also something called black dwarfs. Um, so let's look at these one by one. पहले brown dwarfs को देखते हैं. So brown dwarfs, they're actually failed stars. So if you look at this figure, you'll find out that this is our sun, this is Jupiter, and you look at this object, and you'll find that okay, it does not look like a planet, and it appears that it could have been a star. You will look at the size of it, and you feel that okay, maybe it's not big enough to be a star. And that's not the only reason. Actually, the thing is that it cannot sustain any nuclear fusion reaction, right? So it cannot sustain, when it cannot sustain a nuclear fusion reaction, then how can it be a star? Because all stars have nuclear fusion reactions going on within them, right? So the size, first of all, the size is between Jupiter and some small star. So that is the size range for a brown dwarf. And the second thing is that it cannot sustain nuclear fusion reactions, right? So that is a brown dwarf. Now there is also something called a white dwarf, which I have discussed in earlier videos. A white dwarf is something um, that is an outcome of you know uh, a remnant, outcome of the remaining part of the whole life of a star. So, the star starts from a stellar nebula. There's a stellar nebula, right, from which a star will develop, right? Then it will grow, and it will become a red giant, right? It will become a red giant, and then it will have a massive explosion, and there'll be a remnant, there'll be a small part, right, which will be left, that will be called as a white dwarf. So it looks something like this. It is a very dense object that remains after a dying star exhausts its nuclear fuel. So the star, when it becomes a red giant, after some time it will exhaust its fuel, and then it will uh, you know explode, and then it will collapse on its own. Gravity is a better way to put it. But then it will leave a small white dwarf, which is the core, which is very dense. So that will be left out, and that will be called as a white dwarf star. Now a white dwarf star, it slowly cools down, and it becomes a black dwarf. But we haven't found a black dwarf yet. The reason is that the time it takes to cool down a white dwarf star to a black dwarf star is longer than the age of the universe. So the time taken to cool is so long, it is longer than the whole age of the universe, that we do not we do not find it. Right. So obviously we cannot find a black dwarf star, but theoretically it is something that we say that it will be there. Right. So that's all about a white dwarf and a black dwarf. Then there is also something called a red dwarf. Now, red dwarf is a very common type of a star. It is actually a type of a star that is very common in terms of the numbers. So you look at it, it's a small, cool, faint star, and it's a bit cooler than the sun. And you look at the mass of it. The mass is also somewhat 0.052, around half the times the mass of sun, and it can last trillions of years. And the most important example of a red dwarf is Proxima Centauri, which is the closest red dwarf to Earth. Right, so a red dwarf can have planets around it. Okay, so you look at this and you find that it's much bigger than a brown dwarf, so it's a proper star in that sense, right? And it makes up around 70 to 80% of the stars, so it's very, very common, right? As I told you. So it's very, very common in the Milky Way galaxy, and it is potential for habitable planets. So there might be planets evolving around this, which might be habitable. That is the understanding of a red dwarf. All right, so that's all about the space tech part of it. Right, let's now come to the biotechnology section, and let's look at these things one by one. Um, so let's first start with one day, one genome. So this was in news because it was launched by the Department of Biotechnology and Biotechnology Research and Innovation Council on its first foundation day. Right, so they launched this, initiative, One Day One Genome. Now, this initiative is about public release of an annotated microbial genome. So, microbial, mostly they'll be releasing a bacterial genome. So, bacteria and other microbes, they're also helpful in a lot of ways, right? So they could be harmful, but they're also helpful in a lot of ways, in protection of environment, in protection of human health, right? So to enhance the accessibility of these genomes for research, so you can do more research and maybe you can come up with some solution related to climate change or human health. So that is why they are saying that every day they'll be releasing a bacterial or microbial genome in public, so that you can do better kind of research on them, right? So it will also showcase the unique bacterial species that are found in India. So India specific initiative, right? So what is microbial genomics? It studies the complete genetic material of microorganisms to understand their structure, function, evolution, and interactions. So basically, they're talking about what a genome is. Genome is the complete genetic sequence, right? The complete genetic code of an individual, right? That is what a genome is. So you take the entire DNA sequence and you map it, and that is what you'll term it as a genome, the entire sequence, right? Um, and of course, you know what are microbes? They are organisms like bacteria and algae. So that is the initiative, One Day One Genome initiative. Let's come to another thing which was in news, which is exosomes. Um, so there is a technology called Safe ExoCas. If you remember CRISPR-Cas, you might be finding that to be somewhat uh, the same as Safe ExoCas. So this is also related to gene editing, right? So here what have scientists from Columbia University done? They have used exosomes to deliver gene editing tools, to deliver gene editing tools, right? For obviously for gene editing, right? So what are exosomes? First of all, let's understand what are exosomes. Now inside a cell, this is a cell. Now inside a cell, there are very uh, small sacs, small no sacs which are called exosomes. Now these were initially thought to be uh, carrying cellular waste. They were thought to carry cellular waste, and they did not have a lot of function uh, associated with them. So earlier it was thought that they carry most of the cellular waste, and their cell wall is so um, thin they can easily pass through cells, right? So they can easily, if this is an exosome, it can easily pass through the cells, it can easily enter the cell. That is the understanding. Uh, but slowly it was realized that maybe these exosomes have a larger function also. They are also used for communication, and they are also used for communication, or you can say intercellular communication, right? So they might be used for even that. Now their ability to pass through these cells is what the scientists are now using for delivering uh, drugs or even gene editing tools. So what could be the gene editing tools? Your guide RNA and your enzymes, right? So remember, if you remember CRISPR-Cas9, so something similar, you will pack it up and you will try and deliver it using the exosomes, right? So um, this is the meaning of exosomes. They are naturally occurring vesicles which are potentially used as drug delivery vehicles, and they are membrane enclosed structures which are released into the extracellular space, right? So you can use them for delivering drugs or for delivering gene editing tools, right? So this concept was in news because of this new platform that has been developed by scientists from Columbia University. All right, so let's move on and let's look at the next thing, which is organ on chips. Again, this was in news. Um, okay, so organ on chip technology, basically, uh, they are saying that it will help us achieve Bio E3, which means biotechnology for economy, environment, and employment policy goals by personalizing medicine. So what will it do? How will it personalize medicine? Is that they actually, what they do is they have a chip, or you can say a tray, in which they grow some primary human cells. They will grow some primary human cells, right? And when they grow it, they'll either grow it into a tissue or an organ, right? And then they will also develop all the associated pathways. So what do I mean by that? I mean that they will also build several channels, chambers, membranes, right? Um, so that this will resemble, right? This setup will resemble an organ basically on a chip, right? And then they will test, they might test the efficacy of a drug on this. They might test whether you know the uh, drugs could be delivered in this manner, right? Or impact of any medicine. So they could do a lot of things on this setup, right? So the setup is called an organ on chip, right? Or they're also called as microphysiological systems. They're also called as microphysiological systems, right? So it's a micro scale system that mimics the human body environment, right? So you mimic the human body environment to make it um, suitable for growth of tissues, right? And then you use 3D culture models to recreate organs and diseases. So you will recreate an organ and you will try and recreate a disease, and you will try and use a drug to solve that disease, and you will find whether it is whether it is efficient or not, right? And you'll also control cell movement and behavior via different various chambers, channels, and membranes. You can see a little diagram over there, right? Um, so you'll try and do this. Right, so it works by growing cells on a chip into 3D structures using a polymer resembling real tissue. Tiny fluid channels, they will stimulate blood flow, oxygen delivery, and nutrient transport. Right, so they'll simulate these conditions on a chip, and the goal is that slowly, maybe like right now, you can do it for organs. Slowly, what you will try to do is you will want to recreate the whole human body on a chip, right? So you can do it on a more personalized level, right? You can try and recreate the whole human body on a chip, and then you can maybe test it for uh, the entire body system, right? So this is the understanding of this organ on chip technology, which was in news, which has been in news because of the reason of Bio 3E3 policy, right? So let's look at the next thing, which is mitochondria. So mitochondria has been in news because of Parkinson's disease. So what happens is that researchers have found that DRP1 protein, now this is a protein, um, right? If you inhibit this protein, they feel that it could restore mitochondrial function and it could help treat Parkinson's disease, right? So first of all, let's understand what is DRP1 protein. Um, So DRP1 protein, it plays a role in mitochondrial division for mobility and quality control, right? So if they are playing a role in mitochondrial division for mobility or for quality control, now if there's a lot of DRP1 proteins, then there's a lot of division happening, and that could mean that a lot of cells could die, right? A lot of cells might lose energy because mitochondria is responsible for energy. So they might as well lose energy and they might as well die. And this is exactly what happens in Parkinson's. In Parkinson's, the brain cells start to die, right? So what they are saying is that if you inhibit this protein, then maybe this part will not happen, right? This will not happen, and then this could be a treatment for Parkinson's disease, so the cells would not die, right? So that is why it was in news. And if you like to know more about mitochondria, it is the powerhouse of the cell. It generates energy through aerobic respiration. It contains mitochondrial DNA. So there is also something called mitochondrial DNA, which is around 1%. Uh, right, it is less than 1%, but it is mostly inherited from the mother, right? It is mostly inherited from the mother because it's there in the cytoplasm, right? So if you have some idea about mitochondrial therapies, mitochondrial replacement therapies, you know that mitochondrial DNA is mostly inherited from the mother. So mitochondrial diseases also are mostly associated, are mostly inherited from the mother only, right? So just a little bit of extra information on mitochondria, that they contain some DNA and it can be inherited only from the mother's side. And another related concept is about mitogenome. The mitogenome is just nothing but the entire DNA in the mitochondria, right? It's the entire DNA sequence which is present in the mitochondria. And if you study this, so there's another news item that there was a recent study on mitogenome of South African leopards, and it revealed some insights about their origin and evolution. So even mitochondrial DNA can give you some insights about the origin and evolution of species. All right, so that is all about this one. Let's move on to the next one, which is unified genomic chip. Now, again, here what is happening is that your goal is, first of all, let's look at the goal. Your goal is to enable um, better productivity of Indian cattle breeds. So what you're trying to do is um, basically you're taking samples from the cattle, uh, right? And then you're testing them using a genomic chip. Now that genomic chip is unified in the sense that it has the genetic information of a lot of uh, different species. Now, what you can do is using this genomic chip, you can identify the genetic potential of that cattle, the genetic potential of that cattle. And if the genetic potential is good, if it is good, then what you would want is you would want to breed that particular uh, cattle, right? So, because the genetic potential is good, so obviously you will want to breed that cattle because it will give you higher milk productivity, right? And this will in turn help the farmers. Right, so it was this unified genomic chip is going to help farmers in this way that it is going to help them identify which of the cattle has a higher genetic potential, right? So it will enable genomic profiling and it will enable the genetic improvement of Indian cattle breeds. There are two kinds of chip, Gaw chip and um, Mahesh chip, which is for buffaloes, Gaw chips for the cows, right? And it has been developed by Department of Animal Husbandry, right? Dairy and Animal Husbandry under the Ministry of Animal Husbandry. All right. So that is all about unified genomic chip. Now there's another term that is related to this, which is sex-sorted semen technology. Now here what they're trying to do is they're trying to ensure now sex. If you look at the word sex-sorted semen technology, so they're actually trying to sort the sex uh, of the offspring in the semen itself, right? And they do it in such a way that 90% chance becomes of a female offspring, right? So usually, obviously, there is a 50-50 chance, but then you try to sort your semen in such a way that 90% chance becomes that of a female offspring. So you ensure 90% female calf births via artificial insemination, right? Now, this has been developed by NDDB to boost um dairy production. So, obviously, it is National Dairy and Development Board to boost dairy production and improve breed quality, right? So, this will boost dairy production, it will improve breed quality, it will have an impact on higher milk yield, stronger rural economy, genetic advancement, right? So, all these are benefits of sex sorted semen technology. Okay, let's look at the next one, which is Wolbachia bacteria. Now, Wolbachia is a bacteria which is found in insects, in nematodes and arthropods, and it can be passed only through females. This is a special characteristic of this bacteria, right? That it can only be passed through females. Now, because it can only be passed through females, if it enters a population, it will try and improve the population of females because it survives only in females, right? So that is the basic game of survival. You can say the basic rule of evolution. Now, if it can survive only in females, it will try and increase the population of females, and it will try and manipulate the reproduction processes to increase the female offspring, right? So it will interfere with some of the genes. There's one gene called TRA gene, so it will maybe interfere with that gene, and it will improve the number of female offsprings that are born, right? So what happened was recently it was found that Wolbachia eliminated male wasps entirely, right? Entirely. So they eliminated male wasps entirely in some region, and this is why they were in news, right? Because of this potential that they are only passed through females. Right now, one of the most interesting applications of Wolbachia is mosquito control. Right. So what happens is that in 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 Aedes aegypti, which is major cause of dengue and other diseases, now what happens is that uh, if Wolbachia bacteria are present in the mosquito, Wolbachia bacteria are present in the mosquito, and what they tried was they tried to inject the virus, right, virus inside the mosquito, and they found that in the mosquitoes where Wolbachia bacteria was present, the virus did not grow well, right? Virus did not grow well. Wherever Wolbachia bacteria was present, the virus did not grow well. Now, this is uh, significant from the point of view of the fact that if you introduce Wolbachia bacteria in a mosquito population, right, and especially in the females, and then you let the population, you know, grow for a few generations, and after some time, what happens is that most of the mosquitoes will have Wolbachia, and if they have Wolbachia, then obviously, if the virus does not grow well in these mosquitoes, then how can they transmit it? They will not be able to transmit this virus to the humans. They will not be able to transmit it to humans, and that is how you can bring down the incidence of diseases like dengue. Right. So that is the understanding about Wolbachia bacteria. So that is all about space technology and biotechnology, things which were in news, which were important. Right. And if you have any questions, you can reach out to me, and I'll see you next time in some other video.